Today we're very pleased to welcome Prof. Fairchild over from Oxford, who in fact used to be here in Cambridge, which is where I first got to know him in the Department of Pathology then as a postdoc, working with Herman Waldman, and then the whole group sort of left. And Herman Waldman, as vegetarian, still holds the chair of pathology at the William Dunn and is retiring quite soon. But since that time, Paul has been developing a very strong research interest in the topic for today, stem cells. He's also the founding director of the Oxford Stem Cell Institute, which I've just been hearing has 42 laboratories scattered around Oxford University. That's quite a huge responsibility to keep in touch. And as well as that, actually, worked several evenings a week as a counsellor and in psychopathology, working for the NHS and so on, as it were, in his spare time. So quite how he has time to come over to us as well to speak about stem cells. I'm not quite sure, but here he is. We're very delighted he can come and speak to us on pluripotent stem cells, a medical dream or an ethical nightmare. So thank you, Paul, very much.
Well, it's usually at about times like these in seminars that I begin to deeply regret having chosen quite such a provocative title, as I see you all looking at me expectantly for answers. And maybe I ought to just preface this by just one or two personal comments, because this has in fact been very much a personal journey for me over the last few years, as I've attempted really to try and reconcile my own faith as a committed Christian, but also some of the very deep ethical conundrums which stem cell biology continues to bring about. And actually one event which was most important in my journey actually happened at the age of 18, when I first left home to go and study natural sciences at university, because it was only a few weeks after leaving home that I actually became a Christian. And it was actually a very dramatic event, and it's one that I would never be able to deny or even wish to deny, because it was undoubtedly the most positive thing that has happened in my life. But actually, I just mentioned that because one of the things which I have taken away from that experience is that God has an interest and also places value on every single individual. If I was a recipient of that, then why isn't everyone else also a recipient of his grace and his love and his value? And I mention that because that applies not only to those who are born, but also to those who are not yet born. And of course, that has an important bearing on this whole topic of stem cell biology. I also, secondly, should mention that this is still very much a work in progress. So it's not as though the journey for me has ended. So I would hate to give the impression that I have all the answers, or indeed I have any of the answers. But some of the thoughts that I hope to give might at least perhaps interest you and maybe engage people in discussion and debate. Okay, so as Dennis mentioned, for the last 16 years or so, I've been based in this rather imposing building, the Sir William Dunn School of Pathology at the University of Oxford. And this may in fact be familiar to a number of you here, because it was actually here in this building back in the 1930s and the early 1940s that penicillin was first developed as a therapeutic agent. And of course that opened up the entire area of antibiotics, and the entire antibiotic revolution, if you can call it that. And interestingly, it is that early work on antibiotics which has been at least partially responsible for the current interest in stem cell biology and regenerative medicine. Because it is, of course, the widespread use of antibiotics, together with programs of vaccination, that has been responsible for the increased life expectancy that we have seen over the last 80 years or so. And in fact, so great has been that medical success story that in fact it has had an enormous effect on many populations in the developed world. And just to give you some idea of the sheer enormity of the changes that we can see even before our very eyes, then we need only look at some of these data that were published back in 2006 in The Lancet, which just show you the changes that are happening even in a country like South Korea. Now, as you can see, back in 1960, just a tiny fraction of the population had reached retiring age. And in fact, the entire population was sustained by a large number of children under 15 years of age. But if the current trends in aging continue at the same pace that they are today, then by the year 2040, we will see an almost complete inversion of that pyramid. And what we will find is that up to 25% of the population is retired, and numerically at least, they will far exceed the number of children at school. And this doesn't simply describe South Korea, because you would see very similar patterns in pretty much all of the developed countries at the current time. And in fact, 2006 was an extremely important year here in the UK, because as some of you will know, it was the first year in the history of our nation that the number of people retired actually exceeded the number of children at school. So we've already surpassed that all-important tipping point even here in the UK. Now that is going to have huge consequences. Those consequences may be sociological, economic, perhaps even political, as we've already seen with the pensions crisis. But arguably, it's in the medical healthcare arena that the challenges are going to be greatest. And to give you just a little bit of a flavour of the ageing time bomb, if you like, that is heading our way, let me just share with you a few data that I found recently from the United States. Now, currently, in the US alone, 125 million people, that's about 45% of the entire population of North America, suffer from at least one chronic or degenerative disease. Now, that is set to increase to 157 million people by the year 2020, so we're only talking 8 years from now. Currently, the treatment of those chronic conditions consumes 78% of the entire healthcare budget of the United States, so that you can see as ageing continues that that will very rapidly become unsustainable. But there is one, at least small glimmer of hope, and that is that a significant proportion of these chronic and degenerative disease states could be treated at least relatively easily by the replacement of a single cell type or tissue, that tissue that has become compromised through the natural process of aging. And of course, it is for that reason that stem cells have had such a high profile over the last decade or so, because conceivably those could produce the various populations of cells that we may need to replace in the aging population. If we could only systematically rejuvenate those tissues that are affected through the aging process, then we would effectively have a strategy for dealing with the healthcare issues of the 21st century. And in fact, you may be surprised to hear that a number of different species already adopt that kind of strategy. And in fact, the salamander and all populations, or all members of the Urodeles, have a surprising capacity to be able to rejuvenate many of their tissues. In fact, the salamander is exquisitely capable of dealing with violent combat. And in fact, if it were to lose either its tail or one of its limbs, then it is able to rejuvenate those limbs relatively easily from the severed tissue. And in fact, it can do that on multiple occasions because of the activity of a population of stem cells which is found at the interface here. But in fact, if you really want to be impressed by the ability to rejuvenate tissues, then we need to travel a little bit further back in the phylogenetic tree, back to the planarian flatworm, which actually graced the front cover of Science magazine, as you can see, back in May last year. Now, in fact, the planarian flatworm has been known to be a master of regeneration for quite some time. And in fact, if you take just tiny body fragments from one of these worms, then they are able to regenerate the entire living organism. And that's because of this population of stem cells, stained here in red, known as neoblasts. But in fact, these are no ordinary stem cells, because as Daniel Wagner showed in this issue of Science, it is possible to regenerate the entire living worm from just one of these cells if it is transplanted into a lethally irradiated recipient. And the reason the planarian worm is able to effect this quite amazing feat is that it has been able to harness the property of pluripotency and to entrust it to this population of clonogenic stem cells, or neoblasts. And pluripotency is actually the ability of just a single cell to generate each and every one of the cell types that makes up the body of the adult. So the question that we have to ask this afternoon is, where do we find pluripotency in man, and how can we access that pluripotency in order to use it to treat human disease, particularly those chronic and degenerative diseases I've already mentioned? Well, unfortunately, unlike the planarian flatworm, we don't actually harbour any of these pluripotent stem cells in our adult state. And in fact, we have to wind back the developmental clock right the way back to a period pre-implantation in order to identify truly pluripotent stem cells. Now, this is a human blastocyst about 5 days after conception. Now, the blue area here, known as the trophectoderm, is what gives rise to the placenta and the amniotic sac, all of the plumbing, if you like, of the developing embryo. But here in the centre, these few cells stained here in red are actually stained for a transcription factor known as OCT4, which is actually one of the master switches, if you like, of pluripotency. So it is these few cells which make up what is known as the inner cell mass, which are truly pluripotent. These cells are able to make every one of the 210 cell types that make up the mammalian or the human body. And it is, of course, these cells which go on to form the developing embryo, the fetus, and then the entire individual. Now, unlike the planarian flatworm, unfortunately, pluripotency is maintained only for a very brief period in human development. In fact, it is lost very soon after implantation into the uterus. But actually, a very important breakthrough in the whole field of stem cell biology was published back in 1998 by James Thompson in Wisconsin, when he was able to access this population of cells from human blastocysts that were surplus to requirements in IVF clinics, and to show that those could be maintained long-term in vitro without losing any of that capacity for pluripotency. And these are what we now know, of course, as embryonic stem cell lines. And effectively what embryonic stem cells actually do is to capture that very brief moment of pluripotency that you find in the developing embryo, and then to make it available in vitro, either for studying or, of course, potentially for therapeutic purposes. So in many ways, embryonic stem cells would be the ideal population of cells that you might want to use for therapeutic purposes, particularly to address the chronic and degenerative diseases that we're seeing, those diseases of old age. And there are many properties which actually make these the ideal population of cells. They are, for instance, able to survive indefinitely in vitro due to their ability to self-renew on a regular basis. And this, of course, means that once we have generated an embryonic stem cell line, it is a permanent resource that can be made available to many laboratories or clinics around the world. So there are many people that could benefit from a single embryonic stem cell line that has been generated. And because these can be multiplied and proliferate rapidly in culture, they are able to be scaled up for clinical application. There is at least the possibility that we could make use of embryonic stem cells to address the kind of healthcare needs on the scale that I've already been mentioning to you. And because they can be maintained long-term in vitro, they are, of course, amenable to quality control, which is extremely important when you want to use these cells in a clinical setting. But by far and away the most important property of these embryonic stem cells is, of course, that they are in fact pluripotent. In other words, they will make each and every one of the cell types that makes up the human body. Now, we're able to show that these cells are in fact pluripotent in a number of different assays, particularly in the mouse in this case. You could never perform these experiments for obvious reasons in the human. And this is actually a set of experiments that was performed by Tim Davies in my lab a few years ago. What he did was to take embryonic stem cells, mouse embryonic stem cells, and to genetically modify them to express green fluorescent protein for no other reason so that we could actually trace the lineages that they gave rise to in vivo. What he then did was just to inject a few of those embryonic stem cells into a recipient blastocyst, and as you can see, they actually home back to that inner cell mass, which is effectively the niche from which they were originally extracted. It's then possible to transplant those blastocysts back into the uterus of a surrogate mother, and it's able to implant and give rise to an entire embryo. And as you can see, that developing embryo is entirely green. So what that tells us, of course, is that those very few cells which were injected into the blastocyst have been able to give rise to all of the tissues, or contribute at least, to all of the tissues within the developing fetus. And on some occasions, they will also contribute to the primordial germ cells, which will, of course, give rise to the gametes. And that means that those genetic modifications, in this case, the introduction of the green fluorescent protein gene, can then be transmitted to all of its offspring. And that is the most rigorous proof of pluripotency that we have at our disposal. Now, of course, you couldn't perform these experiments in man, but we are able to show that human embryonic stem cells are also pluripotent by using a surrogate assay. And that is what is known as teratoma formation. If we were to inject just a few of these human embryonic stem cells under the kidney capsule of an immune-compromised mouse, then they are able to proliferate to form a benign tumour known as a teratoma, which contains all of the different tissues derived from each of the 3 embryonic germ layers. If you section through this teratoma, you can find a whole range of different cell types and anatomical structures, from respiratory epithelium in this case, to cardiac muscle, even epidermis of the skin and glandular tissue. And importantly, these are derived from the endoderm, mesoderm, and ectoderm, respectively. So there are 3 embryonic germ layers, so clear proof that these human embryonic stem cells are also pluripotent. And what is perhaps most important about these embryonic stem cells is that they retain their pluripotency even in vitro. And in fact, we now know how to direct the differentiation of these cells into a whole range of different cell types, from gut epithelium on the one hand to spontaneously beating cardiomyocytes to dopaminergic neurons, even populations of leukocytes, so the blood cells. And these again are derived from endoderm, mesoderm, and ectoderm respectively. So they retain that pluripotency even in vitro. But perhaps most importantly of all, many of the cell types that we have learnt to differentiate in vitro could potentially be useful for therapeutic purposes. And in fact, over the last few years, there have been many publications showing the efficacy of using embryonic stem cells, either human or from other primate species, in treating animal models of human disease. So dopaminergic neurons differentiated from monkey embryonic stem cells have, for instance, been quite efficacious in treating many of the symptoms of Parkinson's disease. In fact, it's now possible to use human embryonic stem cells to produce insulin-secreting cells, which are responsive to the external glucose concentration, and that, of course, is a prelude to the treatment of diabetes. And also, it's possible to use those beating cardiomyocytes that I just showed you to actually improve some of the symptoms of myocardial infarction in large animal models. And in fact, they will even act as biological pacemakers if they're actually implanted back into pigs, for instance. And of course, this is potentially just the tip of the iceberg, because being pluripotent, a single embryonic stem cell line could conceivably generate a whole range of different cell types and tissues, which could, of course, be applicable to the treatment of a very wide range of different diseases in humans. And this, of course, was the vision of Geron Corporation, which was really one of the preeminent companies in the stem cell field. And you may already be aware that they undertook the very first in-human trials of human embryonic stem cells for the treatment of spinal cord injury. And in fact, that trial started in 2010. And so far, about 3 or 4 patients have been treated who have acute spinal cord injury with oligodendrocyte progenitors that have been differentiated from human embryonic stem cells. Now, you might have already heard as well that unfortunately those trials were stopped just before Christmas, largely because of the huge costs involved in mounting them, which Geron unfortunately couldn't sustain in the current economic crisis. But of course, where Geron have failed to deliver, other companies are very happy to step into the breach. And just a couple of weeks ago, a paper was published in The Lancet describing how Advanced Cell Technologies, the company that has undertaken this clinical trial, were able to treat a number of patients with macular degeneration of the eye. And in fact, this reported just the early results of that prospective clinical trial and showed that a number of those treated actually had improved visual acuity in the eye that was treated compared to the one that was not. So clear evidence that embryonic stem cells, human embryonic stem cells, may one day actually have beneficial effects in the treatment of human disease. And although it's still very early days, of course, I think this gives us some hope that we may in fact be just on the cusp of actually beginning to reap the benefits of upwards of 14 years of research into embryonic stem cells since they were first derived. So the likely benefits of stem cell biology and regenerative medicine in the future are likely to be very substantial. I hope I've already persuaded you that the magnitude of the need for cell replacement therapies is simply immense and continues to grow due to the aging nature of the population. Quite apart from the alleviation of human suffering that this might bring about, regenerative medicine is in fact extremely big business as well, with predicted sales in the UK of upwards of £3 billion by the year 2014, so just 2 years from now. Regenerative medicine therefore has significant implications both for the health of the nation, but also for wealth creation. But of course, there is a very important cost, which is where all the ethical conundrums come in. Because there is simply no getting around the fact that in order to access pluripotency, and in order to make use of it in vitro, we have to destroy the very embryo from which those cells were originally derived. And that, of course, raises very important ethical questions, which are often very difficult to answer and which touch, sometimes rather uncomfortably, on what it means to be truly human. And that question was posed a few years ago on the front cover of The Independent. And of course, it is your answer to this kind of question which will determine what your view is of this brave new world of regenerative medicine that is opening up. And it is your answer, or the answer to these kind of questions, which in a way has split or divided public opinion over the last 10 years or so. And in fact, there were hints that there was of that divide in the public just before Christmas, as some of you may remember, because it was before Christmas that the European Patent Office made the directive that in fact no patents that involved either the creation or, for that matter, the use of human embryonic stem cells should be awarded. And that was because it was considered that the use of human embryonic stem cells was contrary to what is known in the trade as ordre public, which actually means just general morality. And it was considered that no individual or any organisation ought to be able to profit financially from something that was considered to be immoral. But of course, nowhere has that divide in the public been greater than in the United States, as you may already be aware. And in fact, there are many advertising campaigns which are often very emotive on either side of that moral divide. But this whole field has also been extremely divisive in the political arena as well. And in fact, that began back in 2001, as many of you may remember, with that now infamous decision by the Bush administration, which was announced only days before 9/11, in fact, if you may remember, that federal funding could only be used for the use of embryonic stem cells that had already been generated prior to that date. And no federal funding could be used to generate any further embryonic stem cell lines. Now, importantly, what they didn't do was to prevent the use of private funding for the derivation of human embryonic stem cells. And in fact, this led to the rather bizarre situation in which scientists who wanted to continue pursuing their research had to set up two quite distinct and parallel laboratories, one funded by the federal government, one funded by private investment. And they had to demonstrate that no reagents, no resources, and no personnel were shared between those two laboratories. And were actually subject to continual audit to make sure that no federal funds were actually misused in the derivation of new embryonic stem cell lines. Now, of course, there was some hope that with the appointment of Barack Obama, that the situation may well improve, particularly because of his outspoken praise for stem cell biology. But in reality, we know that that simply isn't the case, and in fact it has become, if anything, even more complex. You may be aware that in the middle of last year, there was an injunction that was issued to James Shirley, which actually closed down all embryonic stem cell research in the United States for 10 days until it was actually overturned in the courts. Now, in reality, that Shirley versus Sebelius case continues to grumble on, and it will actually be heard in the appeals court in April. So still, even at this stage, there is no clarity in the whole field of stem cell biology, and that, of course, has introduced some level of paralysis and uncertainty into the field. But clearly, this is not an optimal situation for us to be in. So what general principles can we try to derive which might enable us to bring down some kind of conclusions about the use of embryonic stem cells? Well, I think it's fair to say that the moral dilemma actually exists only if we view the embryo as a human being in its own right, with the same moral status that we ourselves enjoy. And yet I think it's probably fair to say that the majority of people, particularly in this country, would not actually subscribe to that view. They may, of course, afford or wish to afford the embryo some level of respect, but they would suggest that it has something less than full moral status and human rights. And the premise is based on the conviction that the embryo actually acquires uniqueness, individuality, and humanness, whatever that may be, during the course of ontogeny, but not at the point necessarily of conception. And people who would adhere to this kind of argument point to a whole number of different milestones, if you like, during development of the embryo, where they would suggest that true humanness actually comes into being. Some have pointed to the biological limit of twinning as being a very logical point at which to view the actual developing embryo as a discrete human entity in its own right. And there is, of course, some logic to that, in that the point at which it can no longer separate into two distinct individuals might be regarded as the point at which it becomes truly human and an individual. Others have suggested that gastrulation may be an important milestone, but most usually people would point to neurological development and the acquisition of senescence as being the critical turning point in the development of the blastocyst and the embryo, at which they truly become human. And actually there are many people who would adhere to these different arguments, and their arguments are actually very persuasive at times, but I would contend that actually conception is by far and away the most logical point at which an individual is actually made and becomes fully human. And I would contend that in fact the burden of proof is on those that would adhere to these kind of landmarks as being important to actually show that conception is not the point at which humanness first comes into being. And for those of us who would look to Scripture for some kind of wisdom on these kind of issues, then I think it would also be difficult to contend that in fact conception is not an important turning point or milestone in the development of the individual. Now, of course, the Bible talks an enormous amount about life after death, but it also hints at life before birth. And in fact, it suggests that humanness is something very transcendent, that it is not bound in any sense by time, and in fact it extends before birth and after death. And in fact there are many different scriptures that we may look at, of course, which would suggest that that is the case, but this is perhaps the best one that I know of, and is perhaps the most poetic description of gestation that I certainly know of. For you created my inmost being, you knit me together in my mother's womb. I praise you because I am fearfully and wonderfully made. Your works are wonderful, I know that full well. All the days ordained for me were written in your book before one of them came to be. This suggests that we were actually known by God even before the point of conception, and that we were valued by him from that point onwards. And that, of course, is extremely important when we consider this whole area of stem cell biology. But even if you don't adhere to this whole issue or this idea of life before birth, and that conception is the most important point in the development of the human being, and it's then that humanness comes into being, then nevertheless it is hard to argue that blastocysts do not at least have the potential for individuality and for human life. For the simple reason that if we extrapolate backwards, then each and every one of us was derived from a blastocyst. And as we extrapolate backwards, I suspect that none of us would come to a point at which we would be very happy to relinquish either our full moral status or our human rights. So the blastocyst, I would contend, is as much human and has as much potential for human life as the embryo, as the fetus, and the full-born child. And in fact, that point was driven home to me very forcefully just a few weeks ago when I discovered this particular program known as the Snowflakes program, which some of you may have come across in the United States. What the Snowflakes program does is to try and find adoptive parents for embryos which have been cryopreserved and are surplus to requirements in IVF clinics. And in fact, they have been able to give rise to 250 or thereabouts babies over the last few years by finding those adoptive parents. And those are embryos which would otherwise have been destroyed. And of course, when you see the full-grown baby, it is very difficult to conceive of then destroying that blastocyst in order to remove embryonic stem cells. But of course, 250 children is just the very tip of the iceberg when we actually think of the number of embryos that are produced through programs of in vitro fertilization throughout the world. There are estimates of 400,000 frozen embryos at the current time in the United States alone. And that is, of course, an extremely important point in this whole debate, because it is the inescapable context, if you like, in which stem cell biology is actually practiced. And it gives rise to what we might call the all is or the nothing is lost argument, which is actually the one to which I myself would adhere. So embryonic stem cells are, as I've already described, derived from supernumerary blastocysts from IVF clinics. Now, of course, the The ethics of IVF is something which could also be easily debated, but is probably beyond the scope of this particular lecture. But to suffice it to say that, of course, IVF is something which is legal, which is practiced throughout the world at the current time, and which is actually accepted widely by society. And you very rarely, as far as I'm aware, come across any criticisms of IVF at the current time. So that is the context, if you like, in which we have to consider this whole debate. And there is, in fact, a legal requirement in many countries around the world, including our own, that those embryos that are not implanted and are no longer required should be destroyed after 5 years of cryopreservation. Under those kind of circumstances, it might be considered ethically questionable not to derive some kind of benefit from the inevitable loss of human life. And given that the use of supernumerary blastocysts for ES cell derivation doesn't actually influence in any way the fate of those embryos, which would be destroyed in any case, then it might be argued that there is actually a Christian imperative to pursue life-saving treatments through the generation of embryonic stem cells. And of course, there are many people that might actually benefit from these kind of treatments, as I've already hinted at. My own mother, for instance, suffers very badly from macular degeneration and could conceivably benefit from that treatment that was published in The Lancet only a few months ago. But actually, as director of the Oxford Stem Cell Institute, I receive emails and letters quite literally on a weekly basis from people whose prognosis is far worse than macular degeneration. I could have brought any number of emails with me, but I'll read you just one of those that I received a few days ago. It reads just simply, "Dear Dr. Fairchild, my name is Katerina, I am 27 years old and I live in Lithuania. 7 years ago I had a car accident and broke my spine. I was operated on after 3 days, but as it turned out later, the operation was unsuccessful. If you can, please help me to walk again." And as I said, that's just one of many of the emails that I receive on a very regular basis. And so I would argue that to fail to derive good from the inevitable loss of human life, if those embryos are to be destroyed anyway, when it is actually within our power to do so, is not in its own right morally neutral, and is itself ethically questionable. But it was, of course, that very stance that was adopted by the Bush administration those few years ago, which has been seen by satirists around the world to be essentially flawed and largely illogical. So for President Bush to actually prevent the use of embryos for potentially life-saving treatments on the pretense that we must protect the sanctity of human life, when the very embryos from which those cells would have been derived had already been slated for destruction, has been seen by many people to be wholly illogical. But there are, of course, a number of counter-arguments to the use of these embryos in stem cell research, even if they are already slated for destruction. The first is that by sanctioning the derivation of human embryonic stem cells, we may merely increase the demand for embryonic stem cells and could easily inspire others to perpetuate the destruction of human embryos. And this is, of course, a very reasonable criticism of the nothing is lost hypothesis. But I would argue that no matter how much we might inspire stem cell biology around the world, we will never come close to using all of those embryos that are produced that are surplus to requirements in IVF clinics. And if in fact you see the derivation of embryonic stem cells as a way of mitigating to some extent the evil, if you can call it that, of embryo destruction, then you might argue that you would prefer to have every one of those embryos used for potentially life-saving research. But the other counter-argument is that to pursue the clinical application of embryonic stem cells may in a sense be the thin end of the wedge and could lead to far greater evils, such as human cloning, to overcome immunological rejection of those tissues. And as an immunologist and a stem cell biologist, the one issue which is still likely to derail the whole of regenerative medicine is actually the immunological rejection of the tissues we differentiate from those cells. So it is truly a really important issue that needs to be overcome. And actually it's probably worth just taking a few moments to consider this whole area of human cloning. And for those of you who are not aware of therapeutic cloning, then let me just tell you the actual basic scheme that many people would follow. what we would do is to take oocytes from a willing donor and to remove the haploid nucleus to leave just the cytoplasm, and then to replace that with a diploid nucleus from one of the patient's own somatic cells. Now, in fact, at this stage here, that nucleus is reprogrammed back to a state of pluripotency by factors which are present within the cytoplasm of the oocyte. And so, in effect, this construct is very similar to a fertilized egg. And in fact, if we stimulate that construct either chemically or often with an electric pulse, then it is able to go into cell division and eventually to form a blastocyst, which is effectively a cloned human blastocyst and will share all of its nuclear genes with the patient themselves. And of course, we could use this blastocyst in much the same way as we do those that are surplus to requirements in IVF clinics for the extraction of the inner cell mass and the formation of these so-called nuclear transfer embryonic stem cell lines. And these, because they are pluripotent, could then generate all of the cell types which might be required by the patient for the treatment of their particular disease state. And it is argued, because these are genetically identical to the recipient, that they would not in fact be rejected. Well, there are of course numerous problems with therapeutic cloning which are of a scientific nature. The actual availability of donor oocytes is currently far too low for this kind of treatment. The safety profile of the nuclear transfer embryonic stem cell lines derived in this way has certainly not been proved at all. And in fact, there is a high probability that the tissues we differentiate from those would in fact be rejected anyway, because of minor histocompatibility antigens encoded in the mitochondrial genome, which remains the preserve of the donor oocyte, rather than the patient themselves. But the actual main problem with the whole area of therapeutic cloning is that it is precisely this technology, of course, that was used to create Dolly the sheep. If we were to simply take that cloned human blastocyst and to put it back into a surrogate mother, then we could effectively clone a human being. And that is something which is currently outlawed around the world. And in fact, the responses to human reproductive cloning have been almost universally negative. In fact, within only 2 weeks of the publication of that paper in Nature of the derivation of Dolly the sheep, the European Parliament had already called for a global ban on human cloning, seeing it as a serious violation of human rights and offensive to human dignity. The US National Bioethics Advisory Commission suggested that human cloning might undermine important social values, such as by encouraging eugenics or the manipulation of others as objects instead of persons. Leon Cass, who was actually head of Bush's advisory panel on stem cells, called it a blatant violation of the parent-child relationship and a dehumanization of procreation. And Kalata voiced what many people were wondering, which is that cloning might create soulless beings that are merely genetic shells of humans. But there are, of course, many other arguments that could be used against human cloning. Cloning, for instance, allows the production of humans to order in ways to suit our purposes rather than theirs, and clones would naturally become commodities rather than our peers. That could, of course, be levelled at many other of the reproductive technologies as well, even IVF or the generation of so-called saviour siblings. It's not specific to human cloning. But perhaps most importantly, a cloned child is not effectively made in the image of God, but made in the image of man, one very particular man in the case of cloning, and is therefore denied any genetic uniqueness. And it is, of course, a basic human right to have two biological parents and genetically distinct parents. So that is, of course, the response to human reproductive cloning, but surprisingly, the response to therapeutic cloning, the generation of embryonic stem cells for therapeutic purposes, has actually been surprisingly positive. And yet, in reality, there are enormous ethical issues surrounding the use of therapeutic cloning. Because in this case, not only have we destroyed a human blastocyst in order to generate embryonic stem cell lines, but effectively we created it in the first place using this kind of process. And in fact, to generate human embryos for no other purpose than to destroy them in order to remove the embryonic stem cells from them would, I suspect, reduce human life to a very utilitarian level. So there are enormous problems associated with cloning technology. And in fact, we are now in a very ethically precarious situation of suggesting that to take this cloned human blastocyst, to implant it and give it the possibility of life is considered to be a moral outrage, whereas to generate those cloned human blastocysts for no other reason than to destroy them is actually ethically permissible, which to me seems to be a complete nonsense, effectively. Thankfully, much of the problems associated with therapeutic cloning have now been superseded by this kind of technology known as induced pluripotency, which many of you, I suspect, will already be familiar with. And in fact, this is likely to be the future of stem cell biology, because we now know it's possible to take just a few cells from an individual and to effectively reprogram those back to full pluripotency by the introduction of just 3 transcription factors. And these so-called induced pluripotent stem cells are effectively indistinguishable from embryonic stem cells, at least in their pluripotency, and so we could generate from these all of the different cell types that may be required to cure the diseases of that individual. And of course there are many different advantages to this kind of a scheme over and above therapeutic cloning. First of all, we do not, of course, require any donor oocytes, and so importantly, there is no mitochondrial genome from the donor which is left in this scheme. And so the likelihood of rejection of tissues is far less in this particular instance. It's not actually obviated instantly, but in fact it is reduced. But of course we have also neither generated nor destroyed human embryos in this whole process. And that of course suggests that this might be a much more ethically sensible way forward in the whole pursuit of stem cell biology. I actually gave an interview to the Guardian and the Observer last year, in which I actually extolled all of the virtues of induced pluripotent stem cells, but was intrigued to find that the one thing that they hit on in the headline was actually that you do not require embryos to generate these pluripotent cells. So there is clearly still an enormous interest out there in the use or otherwise of embryos for research. Now, although there are a huge number of problems associated with induced pluripotent stem cells, and I wouldn't want to trivialize those in any sense whatsoever, I think nevertheless that there is evidence already that they are going to revolutionize the whole of medicine in the 21st century. It is, for instance, possible to derive these induced pluripotent stem cells from individuals with particular disease states, and then to generate models of disease pathogenesis in vitro. And in fact, this has already been done with a huge number of different disease states, many of which cannot be modeled in animals, such as Duchenne's muscular dystrophy. It is also possible, of course, using this approach, that we may be able to identify the genes associated with some of those disease states, but also to screen larger libraries of potential compounds for drugs that might actually intervene in that whole disease pathogenesis. But it is, of course, the use of these pluripotent stem cells as a potential source of cell types which might actually be reimplanted back into the individual from which they were generated, which is most likely to enable us to move forward in the whole field of stem cell biology, but without all of the ethical quagmire. At least almost. Because actually there are still a few ethical considerations that need to be taken into account, even when using these induced pluripotent stem cells instead of embryonic stem cells. So the property of pluripotency, of course, may confer on these iPS cells the capacity to undergo meiosis and to form viable gametes. Now that raises the very interesting situation that it might actually be possible to produce genetic offspring from same-sex couples, but also if the egg and sperm could be differentiated from the same iPS cell line, and it is currently a big if, and it has not actually been achieved, then the spectre of reproductive cloning is again raised. And in fact, this was pointed out by The Independent on their front cover just a couple of years ago, when they quite rightly pointed out that this would be a much easier way to clone human beings than ever somatic cell nuclear transfer is. Now, thankfully, we're a long way off actually doing that, and so currently, at least, the use of iPS cells is ethically far simpler than the use of embryonic stem cells, and I would suggest is actually the way forward for stem cell biology. So let me just conclude then by trying to draw together a few general principles from some of the things that I've been able to tell you about. First of all, stem cell biology and reproductive technologies will doubtless continue to raise ethical challenges in the future, And I think it's very important for the Christian community to actually engage in that debate. The magnitude of the medical needs of the 21st century are simply immense and create a commensurate potential for the alleviation of human suffering. But no matter how great the potential for good, the destruction of human embryos to meet that need would reduce human life to effectively a utilitarian level. However, given that there is a legal requirement to destroy supernumerary blastocysts from IVF clinics, there is, I would contend, a moral imperative to try and bring good and meaning from that inevitable loss of human life. Not to act as a force for good under such circumstances cannot be considered ethically neutral, but carries moral consequences of its own. But there is nevertheless a moral obligation, I would believe, to pursue the least ethically challenging line of research, which is actually what makes the advent of induced pluripotent stem cells particularly attractive. And on that point I shall end and perhaps open the floor for discussion. Thank you.
Thank you very much indeed, Paul. Thank you. Well, I'm sure there's lots of questions for you, sir. Yes.
It is absolutely amazing, isn't it? In fact, it generates feelings of awe to think that the simple pluripotential cells can be made to develop into nervous tissue, cardiac mass, presumably by relatively simple changes of the environment while they're developing.
Yes, absolutely.
But what do you think is the influence that requires them to do this and directs them to do this? Are you into some sort of field theory like Rupert Sheldrake's what they call morphic resonance, or what is it?
Sadly not, no.
Do you know anything about it?
Well, we do know certainly how to direct differentiation down particular lineage pathways, as I've already shown it. And we know that that's due to endogenous factors within the, particularly transcription factors within the cells themselves, which we need to switch on in order to convey onto them the properties of a particular cell type. And we know that we can do that relatively easily in some cases by actually altering the external environment, as you mentioned. But beyond that, I'm afraid a lot of stem cell biology is actually a black box, and we really don't know quite how differentiation works.
Yes.
It was said that Dolly the sheep grew old and died quickly because she had degraded telomeres. Would this be a practical issue for any kind of donor cell process?
I think it could be, yes. It's an important issue, actually, because a lot of the tissues that we would derive from iPS cells will have shortened telomeres. But in fact, of course, all of the tissues in our body will also have shortened telomeres. So it's not as though we're putting abnormal cells into an individual. They are as you would expect them to be at that stage of development. So I don't think necessarily it'll cause problems, but it is potentially an issue, certainly. It would be in human reproductive cloning, certainly. Actually, it's interesting that in mice, you can clone mice down many generations, in fact, but eventually they become infertile, and that is actually due to the shortening of the telomeres.
Peace, yes.
Can we envisage other problems in future generations of these particular people?
In which particular people? Sorry.
In people who receive treatment with stem cells, can we envisage the problems that might arise in future generations?
Well, that's an interesting point. I mean, for the person that receives the products of stem cell differentiation, there is a fear that those cells could potentially become tumorigenic, that they could in fact form teratomas, which I showed you a little bit earlier. So that is a possibility and one of the potential side effects of this kind of treatment. But for future generations, I'm not sure there would be any natural consequences. Not that I can think of, at least.
Please.
If the person with the disease has an inherited genetic disease, how do you switch that off before doing the induced stem cell treatment?
Well, that's certainly an important issue as well, of course, because by actually deriving the iPS cells from the patient themselves, if it is a congenital disease, as you point out, then you risk simply perpetuating the pathology effectively. So we have to try and intervene in that whole process to correct the actual genetic defect. And that could be, and has in fact been done on a number of occasions, by homologous recombination. So it is possible to take those iPS cells, which are actually surprisingly amenable to genetic modification, and to correct that gene defect before then differentiating the cells from them. Even that actually is quite complex to do, because in fact in changing the or correcting the gene, you produce what is called a neoantigen, a protein that has never been experienced by that individual before. And so effectively you could mount quite a vigorous immune response against it, which would then neutralize the benefit of having put those cells back in. So it is quite complex, certainly, but there is the potential for doing that.
Please. Yes.
You said early on that IVF clinics are relatively uncontroversial. But you did raise the moral problem about producing embryos for utilitarian purposes. I think your argument then is, well, they're there, it's morally more beneficial or better to use them than to not. The problem, there is a moral problem there in their production in the first place.
Yes, potentially, I agree.
Could you repeat the question, Paul?
Well, as I understand it,
yes.
Your issue is that there is a moral problem with the generation of embryos that are surplus to requirements in IVF clinics,
Yes.
whether or not they're used for the derivation of embryonic stem cells.
There's this slippery slope argument that would come into play if their production is encouraged.
I would agree. Yes, I think that is an issue, certainly. But as I pointed out, in fact, that debate is no longer going on. So IVF is now accepted fully into society, and often you very rarely hear objections about the generation of these embryos, even though they are produced in huge numbers every year and are frozen and are surplus to requirements. But that is actually happening currently, whether we like it or not. And so given that that is the situation, then I would contend that it is better to try and bring good from the destruction of those embryos rather than have them destroyed anyway.
But, are the numbers produced massively greater than the numbers required?
Yes, massively greater. Well, it depends which country you look at, actually, because some countries will only allow you to produce the same number of embryos that are reimplanted. So Germany does, for instance, that. And I think you can only generate 3 embryos at a time and then reimplant them. But most countries actually generate vast excesses of embryos and then choose the best of those for reimplantation. So there are generally in most countries numerous embryos that are surplus to requirements. As you saw in that Snowflakes program, about 400,000 are estimated to be currently frozen in the United States.
Yes. No.
Not that this needs another layer of ethical issues, but I was wondering about the ethical issues of referring to the second point on the slide. If we generate either embryonic stem cells or induced stem cells to treat tissue degeneration and things like that, if we then extend human life through that process and we allow people to live to 100, 110, 120 on a regular basis, what about the ethical issues about consumption of resources, filling up the planet, overpopulation, those kind of issues that we're already struggling with in the contrast between the developed and the developing world?
Yeah, it's an interesting point.
Can you repeat the question, please, Paul?
Yeah, so as I understand it, the question is, well, if we are able to treat these kind of diseases and extend human life even further, do we not just simply aggravate the whole problem in the first place of aging? And it's true, it is an issue. I would contend that we are not actually trying in any sense to lengthen lifespan using these kind of treatments, but merely to try and make life more bearable for people with degenerative diseases during their lifespan. But a natural consequence may be that they live a little longer, but of course that's unknown or untested at the current time. But it certainly isn't the intention, but I do take your point that it could actually aggravate the whole situation.
Please.
As I understand it, things like blood and skin are replaced completely over time, and so even now, is it possible that somebody with resources could decide they want to become luminous, like the mouse, and that is actually practical now, is it?
I'm just contemplating being luminous. Sounds rather good fun, actually.
It would reduce traffic accidents in night.
Yes. You'd never be hit by a car, would you? I doubt it. But I can't come up with any reason for not wanting to be luminous, to be honest. But I mean, it's an interesting point, isn't it? But I think it would be hugely difficult to actually achieve.
Please.
One has read reports that it is possible to take stem cells from an individual and grow for that individual, things like new teeth and skin, which bypasses all these ethical problems. Is this nonsense or is this real?
No, that's real, and that's what I was referring to here as the induced pluripotent stem cells. So yes, certainly.
They're there when one is adult, are they?
They're not adult stem cells, but they are taken from the adult. So they are usually taken from just dermal fibroblasts, in fact, and
real.
Switched back. Oh yes, absolutely. We do it all the time in Oxford. So switched back. Sadly, not on the NHS as yet. Yeah.
Thank you very much.
But yes, it can be done.
Yeah.
Please, yes.
How far away are we away from having this regenerative medicine being fully at work, and what are the main obstacles between now and getting there?
So how far away are we from seeing the benefits?
As in from at least having a stable, kind of trusted application of these principles, and therefore not just kind of seeing one or two trial cases.
Well, it is hugely complex trying to bring these kind of treatments to the clinic, as you can imagine. And in fact, Giron Corporation that I mentioned back during the course of the lecture, I have actually worked very closely with. They actually funded my laboratory for 3 years, in fact. And in order just to begin those clinical trials for spinal cord injury, they had to submit 22,000 pages of evidence to the FDA that these cells would not become tumorigenic. And even then, the FDA were not satisfied. So you can see the kind of regulatory hurdles that you have to get over are huge. And of course, the use of induced pluripotent stem cells is even more complex because these have been genetically modified. So usually when you ask the FDA to approve a treatment of stem cell or cell therapies, they find it extremely hard to come to a decision. But if those cells have been genetically modified as well, they just throw their hands up in horror at the thought of actually putting these into individuals. So actually, even the regulatory hurdles are simply huge. But nevertheless, I think we are literally on the cusp of beginning to see the actual integration of these kind of therapies into normal life, as I mentioned there from the treatment of macular degeneration. But there are others as well, which are happening currently.
And do you have a personal opinion or preference on how this kind of system should be set up? Or
How do you mean that it should be set up?
Implementing the method. Because as you stated, there's so many problems with going through the process of applying the method to solve some trials or whatever. Do you have any opinion about how this should be done in a more systematic basis in the future?
Well, I think my only suggestion or my own strategy is that it should definitely be done by somebody other than me, because it is hugely complex. And by and large, I think the only people that can actually bring this into the clinic are very large pharmaceutical companies, people like Pfizer, for instance, that have the resources to be able to pay for the kind of clinical trials that are necessary. And unfortunately, it is hugely expensive. And that was actually the reason that the trial that Giron started in 2010 was halted, but it was just simply the financial problems.
Please, yes. Yes, that's...
My question comes from a non-scientist, and maybe just an observation rather than a question, because your cartoon of George Bush brought to my mind the whole world of political manipulation and slogans and the manipulation of people's emotions. And I'm wondering to what extent you feel we have right now any scope for the kind of rational, balanced analysis that you have presented both in policy here and in other countries.
Well, I think the debate is a lot more vigorous in the United States than it is here. And often in this country, I don't really find much opposition, actually, to the use of embryonic stem cells or induced pluripotent stem cells, which I think is actually quite surprising. But certainly in the United States, it is very polarized at the current time and continues to be. Quite how one deals with that, I have no idea. I think it'll ultimately be dealt with through the courts, as we're seeing currently. And there will be a ruling by the Supreme Court ultimately as to whether embryonic stem cell research and stem cell biology in general should be supported or whether it should be stopped. And I think it'll be as simple as that.
Last question from, yes.
I was wondering if we understand why in human beings that pluripotency is constrained to such early stages of development, and if there's a way of turning that around, And then also, what is the evolutionary advantage of that, as opposed to other species where that is, as you mentioned, continue to have pluripotent stem cell?
Well, to answer the second question first, I think the issue is actually the lifespan of different species. So for the planarian flatworm, for instance, only has a lifespan of a few days to weeks. And so having pluripotency is actually a major advantage, even in the adult. But there is an important problem with pluripotency, and that is that pluripotent stem cells are just a hair's breadth from becoming teratocarcinoma cells. So for species like our own, which will live into our 80s or so, if we harboured populations of pluripotent stem cells, then the likelihood of tumorigenesis would be exceptionally high. So I think that is actually probably the pressure which has prevented the maintenance of pluripotency into adulthood in man particularly. And I've forgotten what the first question was.
I asked if that could be turned around such that human beings would have pluripotency later.
Well, that is effectively what we've done with this whole area of induced pluripotency, because we know that just by introducing those 3 genes into any cell of the body, we can actually make them into effectively embryonic stem cells with all of the pluripotency of an embryonic stem cell. So we do know how to do it, but why it's not done naturally is another question, I guess.
But if we were salamander-like, it could be quite useful.
It could be.
Regrowth.
And if we were luminous salamanders.
I think it's time to stop on that note. Let's say Paul very much indeed. Thank you so much.